Hello students, in today's session we'll discuss the process of transcription. In the last session we discussed experimental proof for DNA replication, and we also discussed the functions of DNA. We'll recall the functions of DNA. Two very important functions of DNA. The one, DNA can synthesize protein by the process called translation. Okay. By the process called translation, first function. The second one is DNA is heterocatalytic. That means DNA can synthesize RNA by the process called transcription. So what are these? Transcription and translation. The transcription and translation are the steps which are involved in central dogma of molecular biology. So in today's class we'll discuss the first step which is involved in central dogma of molecular biology, that is transcription. Then question arises, what is transcription? Okay, look at here. The process of, in simple terms, the process of synthesis of RNA from DNA is called transcription. Then what is translation? The process of synthesis of proteins from RNA is called translation. Let us start with the transcription. So what is transcription? The process of copying genetic information from one strand of DNA into RNA is called transcription. Okay. So you already know that DNA is made up of two strands. Out of two strands, only one strand which is involved in synthesis of RNA by the process called transcription. The question arises. So when you look at the nitrogen bases of DNA and RNA, during the formation of RNA, it follows complementary base pairing. That means the process of transcription is governed by the principle of complementary, except adenine forms a base pairs with uracil instead of thymine. That means during the formation of DNA, from DNA, adenine base pairs with thymine. During the formation of RNA, adenine base pairs with uracil instead of thymine. The next question arises. So out of two strands of DNA, only one strand which acts as a template strand for synthesis of RNA. Why not two? The reason behind it is, the first reason behind it is, okay, look at here. So the RNA molecule with a different sequence will produce two different proteins. If, for example, if both the strands of DNA can synthesize two RNA, and two RNA, the nitrogen bases are different and they will produce two different proteins that complicate the genetic information machinery. The first reason. The second one is the molecules, the two RNA molecules would be complementary to each other, hence would form double-stranded RNA, and this would prevent being translated into protein. So only one strand of RNA can synthesize protein. If RNA becomes double-stranded, it will not synthesize protein. So because of these two reasons, only one strand of DNA acts as a template strand for synthesis of RNA. Let us start with the transcriptional unit. So the transcriptional unit has three components. Okay. So it has three components. The first one is promoter. The second one is which is located 5' end of coding strand. We'll discuss later what is coding strand. Okay, 5 prime end or upstream end of coding strand. And the second one is structural gene, or we called as a cistron, which is involved in synthesis of protein. And the third one is terminator, which is located 3 prime end or downstream end of coding strand. Okay, this is 5 prime upstream end of coding strand. This is 3 prime downstream end of coding strand. Okay, so there are 3 reasons: promoter, structural gene, and terminator. Let us start with the first one. What is promoter? So look at here. The promoter is a transcriptional unit, is a DNA sequence that provide a binding site for RNA polymerase enzyme. So this is a promoter region where the transcription process starts by breaking hydrogen bonds between 2 strands of DNA. At this site, okay, the RNA polymerase enzyme binds to initiate a transcription. So already I discussed, in order to synthesis of DNA from DNA, which enzyme is required? DNA dependent DNA polymerase enzyme is required. In order to synthesize RNA from DNA, which enzyme is required? DNA dependent RNA polymerase enzyme is required. When this RNA polymerase enzyme binds to the promoter region along with the sigma factors, it initiates a transcription process. Okay, then the terminator, it is located 3 prime end or downstream end of coding strand. So what is the role? It is a terminator is a sequence of DNA that causes RNA polymerase terminate transcription. At this position, okay, what happens? The transcription process ends. Then the question arises: DNA being a double stranded, but only one strand serves as a template strand for transcription at given time. This template strand is called non-coding strand. Look at here. There are 2 strands of DNA. Out of these 2 strands, this strand which act as a template strands for synthesis of RNA, and this strand we called as a template strand or non-coding strand ಅಂತ ಹೇಳ್ತೇವೆ. Non-coding strand or negative strand or antisense strand. Whereas the opposite strand we called as a coding strand. This is opposite strand we called as a coding strand. So DNA dependent RNA polymerase enzyme catalyzes polymerization in only one direction, that is 5 prime 3 prime direction. So the synthesis of RNA takes place 5 prime 3 prime direction on template strand. Therefore DNA strand with a polarity 3 prime 5 prime direction act as a DNA strand with 3 prime 5 prime polarity, okay, which act as a template strand, template and is called template strand or negative strand or antisense strand, while the opposite strand with the 5 prime 3 prime polarity is called a coding strand or a sense strand and coding strand or sense strand or positive strand and the non-template strand is referred to as a coding strand because its sequence will be the same as that of RNA molecule. So the sequences of these are same as in case of RNA. For example, here thymine base pairs with adenine, guanine base pairs with cytosine. So they are complementary to each other. Next, in between promoter and terminator, you find structural gene, or we call it as a cistron. In case of prokaryotes, it is a polycistronic, whereas in case of eukaryotes, it is monocystronic. the structural genes in case of prokaryotes, okay, is polycystonic. That means these structural genes in case of prokaryotes which codes for more than one protein or more than one polypeptide. Hence, in prokaryotes, the transcriptional unit is polycystonic. Whereas in case of eukaryotes, it is monocistronic. That means, okay, the structural genes code for only one polypeptide or only one protein. Is that clear to you? In case of prokaryotes, transcriptional unit is polycystonic. In case of eukaryotes, it is monocystonic. Let us discuss the requirement for transcription. The first requirement which is required is a DNA template. So this is a double-stranded DNA. Okay. So transcription process start at a specific region, and that is called by breaking of hydrogen bond, where the RNA polymerase enzyme binds and initiate transcription process. So out of these two strands, only one strand which act as a template strand, and this strand we called as a template strand, or we called as a antisense strand, or negative strands, or non-coding strands. Okay, non-coding strand. Whereas the other strand, the opposite strand, the strand which is not used for transcription, is called non-template strand, okay, or coding strand, or positive strand and or sense strand. So transcriptional unit, look at here, it has a 5' end of is promoter, upstream end, and in between you find promoter and terminator, you find structural genes we called as a cistron. So you already know that cistron are nothing but structural genes which are involved in synthesis of proteins. And 3' end downstream we find located towards 3' end of coding strand or downstream, which is called terminator. The cistrons occur between promoter and terminator. So DNA dependent, the enzyme which is required for transcription is DNA dependent RNA polymerase enzyme, which polymerase the synthesis of or which catalyze the synthesis of RNA in only one direction, that is 5' 3' end. So this completes the transcription unit. In the next session, we'll discuss the transcription process in case of prokaryotes and eukaryotes. Okay, so thank you.